Good morning, everybody. Today is August 6th of 2024, and we have 50/28th of Ocean's Weekly Seminar and 1/53rd of International Seminar. Today's paper is Nanoplastics in Oral Artificial Intelligence Assisted for the Physicochemical Characterization and Rapid in situ detection. Alicia Law will present today's seminar. Alicia, the floor is yours.
Hello, everyone. Can you hear me?
Yes.
Yes.
Okay. So today I'll present on nanoplastics in water, artificial intelligence assisted 4D physicochemical characterization and rapid in situ detection. First, I'll go through the abstract. For the first time, this paper presented a much-needed technology for the in situ and real-time detection of nanoplastics in aquatic systems. The researchers showed an artificial intelligence-assisted nano-digital inline holographic microscopy, or they call nano DIHM, that automatically classifies nano and microplastics simultaneously from non-plastic particles within milliseconds in stationary and dynamic natural waters without sample preparation. So their results show that the AI-assisted nano DIHM identifies 2 and 1% of waterborne particles as nano or microplastics in Lake Ontario and the St. Lawrence River in Canada. And then they also give some background whereby nano DIHM provides physicochemical properties of single particles or clusters of nano or microplastics, including size, shape, optical phase, perimeter, surface area, roughness, and age gradient. It also distinguishes nano or microplastics from mixtures of organics, inorganics, biological particles, and coated heterogeneous clusters. So this technology allows 4D tracking and 3D structural and spatial study of waterborne nano or microplastics. So I chose this paper because while all of us know quite a lot about microplastics at this point, there isn't much that we know about nanoplastics. This is because nanoplastic research is very difficult. It needs expensive equipment to be used, and it takes a lot of time to carry out, and it's very costly. So I found that this paper was quite interesting since they use AI, and they said that we don't really need sample preparation. So I thought this paper would be interesting for us to read and discuss. So what is nanoplastics? Nanoplastics are tiny plastic particles less than 100 nanometers in size. For scale, nanoplastics are smaller than a human cell, a human, animal, or plant cells, and are in the size range of a virus. Because of their small size, this means that they can spread widely from water, snow, polar ice, air, ground, organisms, plants, and of course, into the human bodies, which poses as a significant health and environmental risk. As of right now, this week, research on nanoplastics and microplastics have been found in the human body, such as in the brain, the blood, breast milk, placenta, and unborn babies, semen, human excretions, and many others, which could lead to physical harm and chemical toxicity. The effects of nano and microplastics on waterborne areas include its possibility to reshape the aqueous carbon storage and contribute to global warming, while nanoplastics and microplastics in the air can alter the Earth's energy budgets and affect climate change. Because of how small nanoplastic is, it is understandable that carrying out research on nanoplastics can be very difficult. So the methodology for this paper was very extensive, not only because they explain quite a lot of things in the paper, but also their supplementary materials, which is 39 pages long, is also part of their methodology. So instead of going through all these technicalities and processes that the paper states, I broke this very long methodology into these five main parts, sorry. So these 5 main parts explains what is a nano DIHM machine briefly and how it works. And then I'll explain how the samples are prepared in both stationary and moving modes, and then how the researchers integrated AI into their experiment and how they validated their techniques. And then I also added this last part, which is model distribution of nanoplastics, because they also discuss on the distinct distributions of nanoplastic and microplastics in a river called Fraser River. So this explanation is going to be very brief. So if you are curious to read more or learn more about it, please refer to the original paper and the supplementary materials. So first of all, Nano DIHM stands for Nano Digital Inline Holographic Microscopy. This means that it can capture holograms of particles using laser light. So here I put figure S1. It shows the figure S1A shows the Nano DIHM instrument, and then B shows the principle of how the DIHM works. So how it works is first a coherent wave from the laser passes through a pinhole, and then from the pinhole, it illuminates the object on the sample holder. Then the reference laser wave and the scattered light form an interference pattern or a hologram on the screen. So C1 shows how it looks like without a sample, so the background of the hologram. And then C2 shows the raw hologram of the sample after it immediately gets lasered by the thing. And then C3 is the resulting hologram of the sample after background subtraction. So after scanning all their data and getting all these holograms, it is then processed using numerical algorithms to reconstruct 3D images of these particles. This method allows for the detailed physicochemical characterization of particles, including their shape, size, and optical properties. So now that you know briefly how nano DIHM works, I'll go straight into how the researchers started their experiment. As you recall, the researchers aimed to carry out experiment in situ, meaning they wanted to test the nanoplastic in the flowing water. But to do that, they need data from stationary samples first. They call this the stationary mode. So nanoplastic, microplastic, inorganic, and organic samples were prepared. This included stuff like PE, PS, PP, PT, PVC, PUR, planktons, oleic acid, metal, chemicals, and etc. And with this, they scanned the sample with the nano DIHM and collected all the holograms of these samples. So once they obtained this data set, these holograms were then fed into a software called the Stingray software. From there, the objects are then found automatically by flexible analysis criteria, and more than 20 morphological parameters, such as shape, size, and etc., were identified. So each particle was then quantified and stored into their database with an assigned taxon. So from there, the researchers trained the AI using this flowchart here in figure S3 of the automated particle characterization and classification flowchart to classify each and every particle scan. So now that they have their stationary database ready, they went and got their moving samples, which they called dynamic mode. These samples were taken from different water bodies of Lake Ontario and St. Lawrence River in Canada. They collected data using 6 pre-sterilized jars while wearing full suits and using gloves to prevent contamination. Actually, in the paper, it wasn't very clear if they actually carried out the experiment there directly, but it's assumed that they did the experiment there. So now that they carried out the dynamic data mode collection, this is where the samples are put into they experimented on these samples based on this diagram. So what they do is that The samples were put on one side of this special flow pipe, and then this flow pipe is placed under the screen of the nano DIHM. One side of the withdrawal syringe pump is pulled, which then replicates the movement of the water on the screen for the nano DIHM to scan while the samples are moving. So with both the data of the stationary and dynamic samples, an auto-characterization table of 6 types of commonly used plastics were tabulated. They used this to teach the AI to differentiate each particle from one another and to help identify which is which particle. And then while the AI is learning and processing this, the researchers also carried out other validation techniques to see if this table was accurate or not. So they used an instrument called the HR-TEM to validate the particle morphological observations acquired. So this is for the stationary samples. And then this machine looks like this here in the middle. And then this machine can zoom into the particles super clearly and very, very zoomed in. So they use this usually to see the cells in the body. And then the next one is that they did use to validate the data from the detection of nanoplastics and microplastics in environmental waters. They use this NALDI-TOF-MS, which is this machine. So these two machines are actually the same thing, but with different functions. And now moving on to the 5th one, which is the distinct distribution of nano microplastic by model. So they used a numerical model called ChemSim3D, which simulates the transport of nano-sized and micro-sized PET plastic particles in Fraser River. So this 3D particle tracking model relies on the Telemec modeling system, which is a software to compute the water depth and horizontal velocity to incorporate the hydrodynamic characteristics of the studied domain. So this model is driven by the fluvial flow at the upstream boundary and a tidal current prescribed at the downstream boundary of the domain in the Straits of Georgia. So this model can capture the stratified flow generated by the freshwater fluvial discharge in the Fraser River. So after this model was developed and prepared, excuse me, researchers carried out a test for both nanoplastic and microplastic transportation in this area. So 100,000 particles were uniformly distributed and released at the water surface at the upstream boundary of the computational domain. The transport of these particles within the system was then simulated for 17 days. Now moving on to results and discussion. We start with the results of the detection of nanoplastics and microplastics. So figure 1 here demonstrates that nano DIHM can detect PE nanoplastic and microplastic within a broad size range from 197 nanometers to over 4 millimeters in water. The intensity and phase reconstruction, such as images and crosscuts of different particles, exhibited unique and distinct characteristics, unveiling particle size, shapes, homogeneities, heterogeneities, and coatings. So if you see figure 1 here, figure 1A specifically, it shows the intensity reconstruction of a PE nanoplastic. And then figure B is the nanoplastic coated by organic oleic acid in water. And then C is PE microplastic. D is the PE pellet at reconstruction plane. And then E to H are the phase reconstructions of the samples A to D. So here in the paper, they only showed the intensity reconstruction of PE samples. But if you go to the supplementary material, they show other samples like PS, PUR, PUL, and etc. And then next is the results from distinguishing nano and microplastic particles. So figure 2 here shows that nano DIHM distinguishes nano, micro, and non-plastics from the heterogeneous blend of PE, PS, oleic acid, magnetite, and phytoplankton in water. So by comparing the shapes and crosscuts of these mixed particles and reference particles shown in figure 1 and figure S7, we can then see the differences between each particles, like some are more circular, some are smaller, some are more square, etc. So for example, if you see particle 4, here they say it's an oleic acid droplet because of a certain characteristics such as it's circular, it's wavy, and so forth. Well then, particle 1 was determined to be magnetite, while particles 2 and 3 displayed specific features of phytoplankton. Particle 5 and 6, the researchers say are likely to be PS and PE nanoplastic respectively, based on the particle shape and size information. Moving on to the results of automated particle characterization and classification, Table 1 presents the auto-classification output of the same mixture samples seen in Figure 2. So these samples were the 6 samples mentioned earlier: PE, PS, magnetite, oleic acid, and phytoplankton taxa. So the results showed that the particle physicochemical properties, including shape, size, perimeter, area, surface roughness, and age gradient, were automatically extracted and compared. However, it was also observed that the size of smaller particles can be more severely overestimated compared to the larger particles. So various size correction approaches were employed to retrieve the accurate particle size information. And then Table 1 provides the particle length and width values with size correction based on previous research. Next is the results from the automated detection of nanoplastics and microplastics in natural waters. On average, 2% of the total particles in Lake Ontario water samples were identified as nano or microplastics. 94% were non-plastics and 4% were unclassified. Among the 452 particles detected in the same amount of the fill blanks, 1 particle was classified as nano or microplastics. The results of a positive control shows a significant increase in the number of particles identified as nano and microplastics. However, there were many challenges to this automated detection method leading to this possible underrepresentation of data. They stated 3 challenges, but I only put 2 here. So the first one is that this method utilizes the shape of the particle to identify plastic chemical composition, and that itself was a challenge because the AI failed to detect other plastic chemical composition due to similar roughness and age gradient values. Another challenge was that the plastic fragments possess irregular shapes such as film, fiber, foam, and granules, and hence were likely to have rougher surfaces, which introduces uncertainties in the auto-classification. So with such challenges, it is understandable that there will be underestimation of the nano and microplastic detection in natural waters. Moving on to the validation of nano-DIHM results with complementary analysis, Figure 3b demonstrates that the morphologies of the waterborne particles acquired using the TEM can be concurrently observed using the AI-assisted nano-DIHM. This shows that the layered and fibrous structure seen by TEM is of a similar shape and size of the particles presented in the manual and automated reconstructions acquired using nano-DIHM. However, it is not suggested that these particles are representing the same particles. So it could be, so these 3 different methods could show the same particle, or it could be a different particle. The researchers are not sure about this either. Next is the results from the 3D physicochemical and spatial data and 4D dynamic tracking. So figure 4 here depicts the 3D positions of X, Y, Z orientations of 13 waterborne particles captured within the studied volume of a single hologram. This particle, the particle dimensions of the 113 particles were plotted in figure 4b and tabulated into figure 4c. Figure 4d are some samples from the dynamic mode, while figure 4e is a compilation of a series on consecutive intensity reconstruction of waterborne particles in dynamic mode, where the particles pass through the water horizontally with the water flow. So these results confirm the results of all these 3D and 4D confirms that the capability of nano-DIHM in detecting nanoplastics as well as its capability to track real-time and in situ 4D dynamics of nano or microplastic particles and non-plastic particles in the water. The results of the distinct distribution of nano or microplastics by model is shown in this figure 5. Figure 5 demonstrates the simulated distribution patterns of nano and microplastic particles within the modeling domain 17 days after their release. Notable difference is observed across the water surface and through the water column. The particle distributions at the end of the simulations are used as inputs in a machine learning-based clustering model to identify the accumulation zones. The results suggest that 33.2% of the microplastics stay within the river domain, while 3.7% of nanoplastics stay in the river domain. So this finding suggests that microplastics have a higher propensity to remain within the Fraser River, while nanoplastics tend to travel greater distances from the point of release, with a significant number of particles reaching the ocean. Now we move to the implication of real-time and in-situ technology. Some of the advantages offered by in-situ and real-time monitoring systems include timely data retrieval, expanded spatial and temporal coverage, possibility to be leveraged to enhance field measurements and data collection, improve accuracy of predictive modeling, and also facilitate effective decision-making. So this laboratory demonstrated that detecting materials in turbid waters is still feasible using nano-DIHM because it can work with minimal light penetration. In situ flow-through sensors can then provide real-time physicochemical data on the quantity and characteristics of nano or microplastics in water, like the AI-assisted nano-DIHM developed in this work. It can also be used to develop scalable monitoring systems for different aquatic ecosystems. Finally, the conclusion of this paper is that AI-assisted nano-DIHM provides quantified physicochemical parameters of nano or microplastics, 3D particle size and spatial information, and 4D dynamic tracking. The development and application of AI-assisted nano-DIHM represents a significant milestone in tackling environmental and health issues caused by plastic. So the researchers of this paper aim to further improve the number density quantification and in-depth surface characteristics to expand the application of AI-assisted nano-DIHM. And then at the bottom here are some additional readings if you are curious to learn more about nanoplastics in the air or by using new methods like microrobots. That's all that I prepared today. I actually have some thoughts, but yeah, that's all. Thank you.